Okay, now that brings us a question here. Why does nature make protein structures so fragile? Protein structure, of course, held primarily by hydrophobic interactions, which are weak. Ionic bonds in water, which are also weak. Hydrogen bonds, which are also weak. All of those things don't take too much energy to break those kinds of bonds. And if you do enough of that, you denature the protein. So, moderate heat can denature proteins. Moderate changes of pH can denature many proteins. I mean, you come down to it, you look at something like drinking a Coke, which has about pH of about 3.5. It's actually quite acidic. It's got, if you look in the ingredients, it's got phosphoric acid in it. Okay, it's quite acidic. This is actually capable of dissolving rust off of a piece of metal. That's acidic enough to denature a lot of protein. If I threw a bunch of proteins in a solution of Coke, many of them would end up being denatured. Okay, well, at any rate, I just had one before I came in this morning. Okay, but at any rate, why do we have protein structure that fragile to begin with? Doesn't sound like it's taking an awful risk that every day we're walking around, we're at the ragged edge of death by protein denaturation. All we have to do is raise our body temperature 10 degrees and we're dead. All we have to do is expose tissues to pHs maybe 2 or 3 units below the normal level or above the normal level, and we start denaturing proteins. Why do we make protein structure that fragile? Why not make them hold it together and stabilize it with tighter types of bonds or more weak bonds? So it's hard to denature proteins. Because after all, it does not take too much, like slightly elevate temperatures to kill most organisms, except for ones that are specially adapted to it. Case in point, when I was a kid, I used to have a freshwater aquarium, and I had a whole bunch of guppies and zebrafish. They're generally pretty hardy fish, right? In fact, zebrafish are widely used as a research organism for genetics and understanding the genetics of development. But anyway, they're also animals' pets. Okay, well, one day the heater malfunctioned. That was long before zebrafish genetics came into play. But anyway, one day the heater malfunctioned. The temperature went up to like 105 degrees. Well, took the heater out, let the water cool down and stuff. Next day, every single one of those suckers is belly up on the top. Okay, slightly elevated temperatures was enough to denature enough of those proteins in those poor fishies that they died and they floated belly up. Okay, so, and then they went to the great aquarium in the sky by way of the massive whirlpool in the bathroom. All right. Okay, well, what happened? Their proteins got denatured and they all died on me. Okay, and back to the local dime store, where we get a bunch more fish, okay, and a new heater. Okay, well, why do we do that? Why do we make protein structure that fragile? Well, there's actually a reason for it. And the reason for it is this: protein regulation. Let's put this baby down here. Proteins are constantly being regulated. Their activities are altered. You have enzymes, for instance, that you don't want active all the time. For instance, imagine our cells have numerous protein digesting and DNA digesting and RNA digesting enzymes in all over, in the nucleus, the cytoplasm, what have you. You can imagine what would happen if those enzymes were active all the time. They would literally chew the cell up from the inside out and kill the cell. So we want to keep those enzymes off most of the time. We want to keep them in storage because we may need them once in a while, but we don't want them always active. And many other enzymes are like that, where we want them active at some times and inactive at others. Many proteins bind to or interact with other proteins to form larger complexes, quaternary structure, or big types of complexes. We want to change that at times. Sometimes we want proteins to interact with their targets, other times we don't. So we have to alter the protein and regulate it that way. We have numerous DNA binding proteins that regulate gene transcription, what we call transcription factors. We don't want those transcription factors active all the time. We want to be able to turn them on and turn them off, so we regulate genes properly. And a lot of diseases, including many cancers, are ultimately caused by misregulation of critical proteins. So, we constantly have to regulate proteins on time scales ranging from days to fractions of a second. Now, how do we regulate proteins? Because protein regulation is critical. Once again, many diseases are caused by misregulation of proteins, where we fail to turn them off when we need to, or don't turn them on when we want to, whatever the case is. That causes all kinds of nasty diseases in humans and animals. Misregulation of various proteins during embryonic development will lead to all kinds of abnormalities, and usually an embryo that's dead at a very early stage of development. Okay, so we have to regulate all these different kinds of thousands of proteins on a constant basis. How do we regulate proteins? We regulate them by altering their tertiary structure, or quaternary structure included. Sometimes we alter it just in a local area, like just around the active site of an enzyme. Other times we may alter it in a more global fashion.
It's just a tertiary structure?
Excuse me?
It's just a tertiary structure? Not
Well, that also includes quaternary structure as well. But if you alter the tertiary structure, quaternary structure may come together or it may fall apart. Remember that example I gave you? Okay, we've got that hydrophobic alpha helix region that sticks to one of its partner. If we bury that inside the molecule, altering the structure, then the binding no longer happens. Okay, so we alter the protein's tertiary and or quaternary structure. That's going to alter, we can either do that in a little limited area of the protein, like say the active site of an enzyme, or we can make massive changes in the entire protein structure, and that can have far-reaching effects all over the place. And then we have to be able to reverse that kind of process. So, we alter the tertiary structure, and we have to undo it, do it, undo it. And then once again, this can happen on time scales of milliseconds, or it can happen on time scales of hours or even days. Many proteins, we have to constantly go back and forth. Switch it on, switch it off, switch it on, switch it off, switch it on, switch it off, far faster than I can even say the words. Now, problem is, if you let's suppose protein tertiary structure was held tightly together. If you want to change that structure, it would take a great deal of energy to do so. And since you're doing this all the time, the energetics of it, the energy usage of that would be too great for an organism to handle. It could use its entire energy reserves just to regulate a limited number of proteins and run out of power. But if it's weakly held, okay, if protein tertiary structure is weakly held, it means you can alter that structure easily, quickly, and not use too much energy to do so. The downside of that, of course, is it's fairly easy to alter to the point that the protein flat out denatures. But protein regulation is so important, it's worth the risk to stay out of hot water and stay out of acidic or basic environments. Then you'll stay away from detergents and you'll be okay.
Let's just live in a bubble.
All right. So that's why we have weakly held protein structure, because we're going to constantly have to regulate these proteins. Now, how do we regulate things? Well, we've seen some examples. For instance, we use the example of changes in pH. That can alter a protein's tertiary structure and make an enzyme more or less effective and things like that. Okay? Now, it turns out, although not all that common, there are a number of proteins that are inside our cells that are pH regulated. Many of these proteins are in compartments or organelles, membrane-bound compartments, and they're deliberately kept at a different pH than the rest of the pH of the cell. For instance, the inactivated. There are other proteins that are going to bind to things at one pH, and then they get shipped into an acidic compartment, and in the acidic conditions, they drop off whatever they're attached to. That's an important cellular mechanism, too. After fertilization of an egg cells prior to fertilization tend to be a little acidic, pH maybe about 6.8. Once fertilization occurs, the pH goes up to about 7.2 in a fairly short order, and that activates a number of proteins involved with the events of fertilization and early development that were inactive even at those slightly low pHs. So we do see pH-regulated proteins. We can also see that various kinds of ions can regulate proteins. Various kinds of small molecules can regulate proteins. And even other proteins can regulate proteins. So there's all kinds of different things inside cells that can regulate protein activity, protein function, by in one way or another altering the protein's tertiary structure. And once again, protein regulation is critically important for cell function, cell survival, and for the organism itself. Screwed up regulation is the of many proteins is the cause of many different kinds of diseases. Okay, now we can break protein regulation down into two major categories. Let's take a look at this, and then I will end the video because then that brings us right to the previously posted stuff. Yes?
You said that it's important for cell regulation, the life of the organism, and what else?
And the function of an organism, the health of the organism.
You said something else about the cell.
Oh, what was that? Excuse me?
You said something else about the cell.
Oh, well, cell function, cell survival. I think that might have been the one, cell survival. Okay, well, there's two basic ways we can regulate proteins. One is by non-covalent, in other words, using weak things like ionic bonds, hydrophobic interactions, hydrogen bonds, combination of them. Non-covalent binding of some kind of regulator, be it an ion, another protein, a small molecule, or whatever the case is. Now, since non-covalent bonds are weak, these interactions are easily broken. The other way we can do this is actually attaching some kind of regulator, which is frequently an ion, to a protein with covalent bonds. Covalent bonds are strong. Once you make them, it's hard to break them again. So, we call this covalent modification. So non-covalent binding, we'll see more on the advantages of this. They're weak bonds. They're easily formed. and they're easily broken. And it's a rapid response kind of process. You can do it, undo it, do it, undo it. So that's really good when you want short-term regulation. Either when you want to regulate something for a brief period of time, that could be milliseconds to minutes, or you're in a situation where you constantly have to regulate the protein. You've got to switch it on, switch it off, switch it on, switch it off, and so on. When we want to do things fast and quickly reversible, that's the way to go. Non-covalent binding of something. Now, covalent modification, where we're forming covalent bonds or breaking them, once you do it, it's stable. It's pretty stable. Most of the time you covalently attach something to a protein, that stuff is not going to break off. It will rarely fall off spontaneously. Now the downside of covalent modification is this. When you form covalent bonds between the protein and some kind of regulator, that's generally a condensation reaction, which is endergonic. It takes a lot of energy. So it takes energy and special enzymes to do that modification, to catalyze that modification. So it requires energy and enzymes to do. Requires energy and special enzymes to actually do that. Now, in order to reverse it, you need a second set of enzymes, because once you form those covalent bonds, they will rarely break on their own. So you have to have enzymes that catalyze the hydrolysis reaction, which breaks that regulator off of the protein. So it requires a second set of enzymes to reverse it. Now, in the case of reversing covalent modification, that happens to be an exergonic hydrolysis type reaction. But the thing is, with hydrolysis reactions, they still need to be catalyzed. They won't occur very much in ordinary body conditions. They will occasionally do so, but most of the time you need to have enzymes to catalyze it. That will not require another source of energy because that's an exergonic reaction. But you do need to catalyze and speed it up. Okay, so you have to have a set of enzymes to do the covalent modification, an energy source to do that, which is frequently ATP hydrolysis, and you have to have a different, entirely different set of enzymes to reverse a covalent modification. Now, the other problem with that is you have to recruit and call to action these different sets of enzymes every time you want to do or reverse this kind of stuff. So that takes time. That's a slower process that may take on the order of seconds to many, many minutes to hours. So it's not something you can do back and forth really, really rapid. And if you say, I want this modification now, you're not going to get it now. You might get a couple seconds later, but it takes time because you have to call out these enzymes to do these reactions. So this is something that is better for long-term modification. Because it takes time to do it, it takes time to reverse it. So we have a set of modifications, a regulation that's good for the short term and good for constantly changing needs. And we have a second set that is better for the long term because you're doing stable changes in the protein. Now these are not exclusive. Many, if not most, proteins can be regulated by both ways for different purposes. So that's what we see with the covalent and non-covalent modification and non-covalent regulation. Question?
What does it say about long term?
Okay, well, it's a long-term covalent modification is best for the long haul because it takes time to do it. Once you do it, it stays that way unless you deliberately call out the enzymes that do the hydrolysis. So it's good if you want to change a protein for minutes or hours or something like that. It's not so good if you need to change things every fraction of a second. If you do that, then you want to go the non-covalent route.
I
Okay?
understand. I was talking about the line above it. I
Oh.
can't read it.
Oh, this? This?
Right above long term. The line.
Oh, this?
Yes.
Oh, requires enzymes to reverse it. In other words, enzymes to do the hydrolysis reaction that removes whatever you attach by covalent bonds.